between two diastromers? Difference between diastromers. They are not the mirror image of each other. They are not the mirror image of each other. This means that the direction of OH group around C chiral group are not the same, and also are not mirror image in the mirror image. However, for two enantiomers, although the direction of OH are not the same, but those directions are completely in a mirror image. For example, whenever OH group is located in the right, the other one is in the right hand, and so on. The OH group, which is located at the right ward, the other one is in the left ward, and etc. Also, we learn about anomeries or anomers, which are the enantiomers, which are the isomers who differ. Sit down, please. Take a seat and sit down. Their difference is about the OH group and the OH group, which is located on the anomer carbon. Anyone can explain what is the anomer carbon? Did you read anything about our previous classes in Iraq? I don't think so.
Doctor, if OH group is up, it's another thing, beta.
Yes, but what is the anomer carbon? Anomer carbon is the carbon which is produced after the those carbohydrates, those sugar, those, for example, glucose, will turn to a from linear form to a circular form. After carbohydrates, for example, a glucose, will transform from a linear to a ring form. We have a new isomers named anomerics. When a carbohydrate, when a sugar transforms from a linear form to a ring form, we have a new carbon, we have a new chiral carbon named anomer. The difference of two ionomer is about their OH group. Whenever the OH group and the CH2OH in the same direction, we name such isomers is in a beta form, and vice versa. If the OH groups and the OH group and the CH2OH group are in the different direction, opposite direction, we have an alpha. But the last isomer is in carbohydrate is, of course, this is sometimes stereoisomers, is epimers. What is the difference between two epimers? The only difference between two epimers is about just one OH group. Just one OH group. If two carbohydrates, two sugars are the same, are a similar structure, but for one OH group around a chiral carbon, we name such isomers as epimers. For example, D-glucose, D-galactose, D-glucose, D-mannose. The difference between D-mannose and D-glucose is about their OH group, the direction of the OH group for carbon number 2. The difference between galactose and glucose is about the direction of OH group around carbon number 4. The only difference between two types of sugars. Finally, the last subject about monosaccharides is about their reactions, various reactions of monosaccharides. The first one is oxidation. As you can see here, when an alkyl group it transforms, it changes to an acid. As you can see here, when an aldehyde is oxide, it changes to an acid group. This is an acid group, COOH. This is an acid group. We have a same process for ketone groups. So, whenever we have a ketone or aldehyde group and we want to oxidize these groups, we then have an acid group. On the other hand, when an alcohol group or OH group oxidize, it transfers to an acid. We also have an acid. You can see here such reaction here. When CH2OH oxidize, this group transfer and change to an acid group, COOH. So, for sugars, for carbohydrates, this is the one of the most important reaction for sugars. When their functional groups, which are aldehyde or alcohol groups, oxidize, these groups change to acids. Both aldehyde and alcohol change to acid. Well, if we have a reaction about aldehyde, just the aldehyde group reacted. So, we have such molecules. We named it aldonic acid. Aldonic acid is a molecule which is produced from the oxidation of carbonyl functional group. What are the most important carbonyl group in nature? Carbonyl group. How much carbonyl group is existed in the nature? Very good, very good. You have two carbonyl groups. The first one is aldehyde, and the next is well. So, if we have an oxidation reaction for aldehyde group, then we have aldonic acid. You can substitute the name of sugar instead of this part. For example, glucose change to gluconic acid. Gluconic acid. Glucose change to gluconic acid. The name of sugar substitutes with the ald part. For example, for galactose, we have galactonic acid. For mannose, we have mannoric acid, and etc. If we have an oxidation process in alcohol group, we name such sugar, such molecule as oronic acid, uronic acid. In Persian, oronic acid, uronic acid, uronic acid. So, uronic acid is a molecule which is produced from the oxidation of carbon number 6. Carbon number 6. Oronic acid. You can substitute the name of sugar instead of or before of this part. For example, for glucose we have glucuronic acid. For galactose we have galactronic acid. For mannose we have mannoric acid, and etc. And finally, if the reaction situations are completely rigid with the presence of acids, both aldehyde and alcohol group are oxidized, and we have a new molecules with both two oxidized groups. Both aldehyde and alcohol are oxidized and changed to such molecules. We name these molecules as aldaric acid. Aldaric acid. So, after oxidation of sugars, we have three types of new molecules. The first one is aldonic acid, the next is uronic acid, and the last one is aldaric acid. For example, for glucose we have glucaric acid. For mannose we have mannoric acid. For galactose we have galactaric acid. you can substitute the name of sugar with this part. Glucose, galactaric acid. Mannose, mannaric acid. Galactose, galactaric acid.
Doctor, can you get it much better?
Yes, you want more explanation? You want more explanation about this? Huh? Well, the first type of sugars, monosaccharide reactions, chemical reactions, is oxidation. Oxidation means a molecule gain oxygen, missed hydrogen, or missed electron. In oxidation, a molecule gains an oxygen, missed hydrogen group, or missed electron group. In contrast, in reduction, a molecule gain hydrogen, gain electron, or missed oxygen. So, such type of reaction are typical reaction of oxidation. You can see that this molecule gain an O group, oxygen group. So, in oxidation of monosaccharide, three different type of molecule will construct or will produce. The first one is oxidation of the first carbon or anomeric carbon or functional carbon, which is the aldehyde group. When the aldehyde group is oxidized, it changes to an acid. CHO change to COOH. This type of molecule named aldonic acid. The next one is oxidation of carbon number 6. We name such molecules as urinic acid. So if the only carbon which is oxidized is carbon number 6, we have a new molecule named urinic acid, for example, glucuronic acid. And finally, if both functional group will be oxidized, we have a new molecule named aldaric acid. For example, for glucose, it is glucaric acid. Glucaric acid. So, three different parts of types of oxidation. The next reaction of carbohydrate. Let me talk about the usefulness of this reaction. What is the usefulness of oxidation in clinic? Well, we use such reaction to identify, to determine whether a sugar exists in a solution or not. As you can see here, sugars can produce oxidized form of them. So, all sugars with a free anomer group. We explain what is the anomer group. All sugars, you can write this, all sugars with a free anomer group can attend can participate in the oxidation reaction. All sugars, not only monosaccharides, all sugars, not only monosaccharides, with a free anomel group. What is our mean about free anomel group? It means the OH group is free and not reacted, not participate in a reaction. Those sugars with a OH group in the first carbon after the anomel carbon. All sugar with an OH group in the anomel carbon can participate in such reaction. So, one of the most important oxidation substances in nature, in clinic, is Cu salts. Cu, copper. Cu salts. For example, tartarat, Cu tartarat, Cu, other salts of other forms of Cu. As you can see here, this molecule can reduce Cu, as you can see here, 2 plus U, 2 plus, change to 1 plus. So this molecule gain an electron. So Cu will be reduced. On the other hand, sugar will be oxidized. Cu oxidizes the sugar. Cu oxidizes the sugar and gain an electron. Cu reduced, sugar oxidized. When Cu reduced, the color of Cu will be changed. The color of Cu will be changed. We will do such reaction in our next practice class today. We name this reaction as Benedict reaction, which is one of the most important reaction in clinical laboratories. Benedict reaction. What is the Benedict reaction? In the presence of Cu salts, Cu compounds, sugars with a free OH group. Please write this explanation. Cu with a free OH group under anomel carbons will be oxidized and on the other hand Cu will be reduced. When Cu reduces, the color of reaction color. So, when the color of reaction changes, you can understand whether a sugar is existed in such solution or not. For example, when a child exerted sugars from his or her stool or his or her urine, and this is like diarrhea. One of the important causes of diarrhea is exertion of sugars in the stool.